Roth column and your minimal medium column with lots and lots of entries. And later ones, the ones towards the bottom, are the newest ones from yesterday. And so when you're doing the analysis, you can get Excel to do all the major, or at least the initial parts for you. It would want to do the mean. Excel calls that average, as you probably know. There are several options for standard deviation. I think this is the one that you want. stdev.p is the version of standard deviation. I think that's probably the best for this analysis. Okay, so when you're looking at the list of functions, that's the one I think you want for standard deviation. Okay. Other than that, I think it's kind of just plug and chug in terms of those calculations. Okay, so that's up there. A couple other quick announcements. I've been asked to do to hold a review session, review in quotation marks, session for next week's test. So I've scheduled that for Wednesday. Common break. Hopefully in this room. I didn't get confirmation on that yet, but we'll assume it's here until I hear otherwise. Review is in quotation marks because I will come and be happy to try and answer any questions that you bring. I can't possibly teach the last 4 weeks of the course again in an hour. So, but if you have questions about what's this going to cover? Metabolism, photosynthesis, DNA, RNA, chromosome structure, and DNA replication. I think that's all the topics. And bring those with you to this room Wednesday at common break, unless I change the room. And I'll try and clear things up. Okay, so that'll be next Wednesday. Okay, so we did finish up DNA replication last class. Okay, so we completed all of the information for the next exam. Remember that the second exam is all Scantron. Okay, so you basically need a pencil to do the exam. So we have 100 Scantron kinds of questions. Okay, so no rest for the wicked, so we have to keep moving. So our next topic, still within this broad section of molecular biology. Now that we know what DNA looks like, now that we know how it copies itself, the next sort of question to ask is, how does it encode information? So just having the double helix was a huge leap forward and certainly worthy of a Nobel Prize, but it doesn't directly answer the question of how genetic information is somehow encoded in that biological molecule. So we're going to turn our attention next to the genetic code, which was sort of the next big thing that people started studying after Watson and Crick had published their structure. So in addition to Crick, so Francis Crick continues to work in this to some extent. We'll talk about his contribution next. But most of the code was broken by these two gentlemen and their labs. Marshall Nierenberg, who I've been out of grad school a long time now, so maybe he's not at NIH anymore. But when I was in grad school, at least, he was still at the National Institutes of Health down in Bethesda, Maryland. And Levine Karani, I don't know where he is. I'm assuming they're both still alive, but I'm not positive of that either. But in any case, these two gentlemen and their labs were primarily responsible for breaking the genetic code, which for the time period was a pretty huge advance as well. If we had the technology we have now and had that problem to solve, it would be not much of a problem. But back in the late 50s and early 60s, when the work was being done, this was completely new territory. They're devising new techniques to try and answer this question. So let's, before we go to the next slide. So, you might not be surprised by the next story I'm going to tell you in terms of Crick's contribution to breaking the genetic code. He continues basically in the fine tradition of Watson and Crick, who don't sully themselves with doing actual experiments. So he contributes a thought experiment to how the genetic code should work. So think about the structure of DNA, right? We have the double helix, sugars and phosphates on the outside, these nitrogenous bases pointing to the inside. And Crick says, well, surely the information can't be contained in those sugars and phosphates, right? It just kind of doesn't make sense. They're just all over the place. They're consistent, so information probably can't be stored there. So it's got to be in these nitrogenous bases, and probably the order of these bases. So that's his sort of assumption. And then the problem that's, if you take that assumption, then the next problem that presents itself is how do we get 4 nitrogenous bases to somehow code for, as we already know, the 20 amino acids that are commonly found in proteins. What's the relationship? So Crick does a little thought experiment. He says, well, if we look at them one at a time, 4 to the first power, then clearly we only have 4 different possibilities, right? B's and C's and G's and T's. And short of trying to come up with some bizarre mechanism where each of the 4 bases somehow codes for 5 different amino acids, which doesn't seem reasonable, you probably don't read them one at a time. So he says, all right, well maybe we read them 2 at a time. We read them as pairs, or not as pairs, but as doublets. So if you do that, then you get 4 squared possible combinations of 2 letters. And that gets us close. That gives you 16 doubles. But we seemingly need at least 20. So 16 is close, but not close enough. So he says, all right, maybe we read them as triplets. We read 3 letters at a time. And if you do 4 to the third power, then you get 64 different possibilities of reading 4 letters 3 at a time. So that gives you at least 20, gives you a lot more, but it seems to be the smallest combination that gives you at least what we need. So Crick says, I think it's a triplet. You're going to find that it's a triplet code. You know where this is going, right? So indeed, by sitting down with a piece of paper, he predicts quite correctly the fundamental, or a fundamental characteristic of the genetic code. God, how do you not hate that? Okay. So, indeed, that is the first major characteristic of the genetic code. It is indeed a triplet code. So, we have to read 3 nitrogenous bases to code for 1 amino acid. And it took a number of years to then figure out which amino acid went with which triplet. And the triplets are usually referred to as codons. And so each triplet is called a codon. So they had to devise new ways of new experimental approaches and all kinds of new technology to figure this out. But eventually, they do assign all 64 codons to specific amino acids. And here's a typical version of the genetic code table that's in pretty much every biology textbook on the planet. Well, very quickly, even from the thought experiment, the next characteristic was sort of implied as well. Since we have 64 triplets, or codons, and we only have to code for 20 amino acids, that's probably going to mean that the code is redundant. And as you can see from the table, it is with 2 exceptions. So redundant meaning that almost all of the amino acids are encoded by more than 1 codon. Okay, so you can see glycine down here is encoded by 4, aspartic acid is encoded by 2. What else is a good one? Arginine is encoded by 6, 4 here and 2 there. So there's mostly redundancy. So there are 2 exceptions to that. Tryptophan only has a single codon, and methionine only has a single codon. So there are exceptions, but by and large, the other 18 are redundant, having 2, 3, 4, or 6 codons available. But if you'll notice, it's a, if not logical, at least a reasonable way of how this is working. It's not random. So it's not like lysine is encoded by GGG and CCG and CUC and UUU, right? It's not all over the place. It's all 4 right there. Okay. So, and that's true for any 4. Arginine are those 4. Serine, those 4. Proline, those 4. So they're always sort of clumped together. And we can see patterns then. So glycine is encoded by GG anything, right? So GG whatever codes for glycine. CG whatever codes for arginine. CC whatever codes for proline. So when there are 4, they're always clumped together as a, sort of a set of 4. And even the ones with 2s, there is a pattern. So aspartic acid is GAU and GAC, whereas glutamic acid is GAA and GAG. What's the pattern here? Pardon? Yeah, GAU, GAC code for aspartic acid, GAA, GAG code for glutamic acid. I'm asking what the pattern there is. Say it again? That's right. So when there are 2, the pattern is the third position is either the 2 pyrimidines, so GAU and GAC, U and C are the pyrimidines. That codes for aspartic acid, whereas GAA, GAG, GA purine codes for glutamic acid. So all of the 2s work that way. Asparagine, AA pyrimidine, lysine, AA purine. So there is some sort of logic, if you will, to how the redundancy is sort of organized in the table. But general idea is it is redundant. Okay, so except for those 2 exceptions, methionine and tryptophan, all the others are encoded by at least 2 codons. But with this idea of redundancy also comes the idea of what most people refer to as codon bias. So the ones that are encoded by multiple, or the amino acids that are encoded by multiple codons, it's rare that all of the codons are used equally. So this is looking at some examples from E. coli. So let's use glycine again down here. So it has 4, but you can see GG pyrimidine is used almost exclusively, is basically used exclusively over the other 2. Even here with the pair, aspartic acid is GAU and GAC, but it uses GAC like almost 3 times as often as it uses GAU. Same thing up here. For threonine, it uses AC pyrimidine more often than AC purine. And not only in bacteria, people have shown the same thing in animals, different biases, but there's almost always bias. So this was discovered, as you can see here, around the 80s or so. Still don't have a great explanation for why. We can show it pretty easily. It's kind of an accepted aspect, but we don't exactly know why they're not all used equally. So there's a great, I don't know if it's a Nobel Prize worthy discovery, but it'll probably get you a doctorate if you figure out what this is all about. So we don't really, like I said, we don't really know why there seems to be codon bias, but there almost always seems to be. So that's just kind of out there as well. We'll get back to that in a second. Let's go back to here. Okay, so we have a triplet code. We have a mostly redundant code. But as they were assigning codons, they found that 3 of them didn't code for an amino acid, but coded for translational stop. So as they're making the protein, and we'll study that how that happens next week, there needs to be sort of a stop signal for the ribosome to know where the information to make the protein ends. So there are translational stop signals, and they're all up here. So UAA, UAG, and UGA are the stop codons. When they found stop, they, of course, asked themselves, well, if there's stops, are there start codons as well? And the answer turns out to be yes. But there's only one of these. Translational stop, or I'm sorry, the translational start signal is always AUG, which, as we go to the code here, sort of does double duty. So it not only is start, it also codes for methionine. So what does that suggest about all proteins?
They all start with it.
Yeah, all proteins must start with the amino acid methionine, which is true. At least as far as initially as they're being made. But as we continue on with how cells make proteins, we'll see that quite often many proteins have the first 10 to 20 amino acids chopped off. And we'll see why that happens and when that happens. So while at least at first all proteins start with the amino acid methionine, they don't always, their functional form doesn't always do so because, like I said, the first few amino acids are often chopped off. Again, so we'll see why and how that happens. Probably not next week, the week after. Okay, so for now, characteristic 3, there are start and stop, translational start and stop codons. Okay, so AUG for start, UAA, UAG, and UGA for stop. Okay, so 3 starts and 1 stop. So that means then that only 61 of the 64 codons code for amino acids, right? Because these stop ones are just stop; they don't code for an amino acid as well. Okay, so far so good. Okay, so we have a triplet code. We have a mostly redundant code. We have translational start and stop signals built in to the code. The next characteristic they thought was always true, but subsequently we have shown that it is not. So we'll call it universal, and then we'll change that, or we'll modify it. So initially, people thought the code was universal. And with small exceptions, it is. For the most part, all cellular life on Earth uses this basic code. But then people found some exceptions. So we hop to slide 6. There were some exceptions. And the list of exceptions kind of keeps growing. So the big exception, and the first one that was discovered, was that the genetic code inside the mitochondria, and subsequently inside the chloroplasts as well, isn't exactly the same. There are a couple of key differences between what's now referred to usually as the nuclear code versus the mitochondrial code. And some of them are shown here. Let's see if we can pick them all out. One is that what is a stop codon in the nucleus, or for nuclear genes, is now a coding codon. It codes for tryptophan. So there's one difference. Are they all serine? No. Okay. AGA and AGG usually code for arginine, but in the mitochondria, they code for stop. So the mitochondria actually has 4 stop codons instead of 3. What else is different? I think the other big difference is AUA, which usually codes for isoleucine in the mitochondria, also codes for methionine. AUG is still start, but AUA also codes for methionine as well. I think those are the big differences. So everything else is the same, so it was what? 1, 2, 3, 4 differences. So not too horrendous, but it was no longer absolutely universal. Right? So there were these slight differences. So, I don't know, nearly universal, if you want to modify it to that. So that was the first big discovery, was that it was a little bit different inside the mitochondria. Again, other people showed similar differences inside chloroplasts. But then other people, and we'll look at this link here, have also found slight differences in the nuclear codes of some organisms. Most of them turn out to be protists, although not always. So this is hopefully the most current list. So now we do kind of technically have to talk about genetic codes, plural, because again, it's no longer universal. But the standard code, the universal version. But mitochondrial, we just looked at, but apparently there's some slight differences between the mitochondria in vertebrates versus the mitochondrial code in yeast. I haven't looked at I must admit I haven't looked at all of these in great detail. But apparently there are differences in mitochondrial codes from different groups. But then the other ones that don't say mitochondrial are nuclear. So there are some differences in ciliates, things like paramecia. I don't know what all of these critters are, though. I don't know what euplatids are. But molds and protozoans, trematodes, that's mitochondrial. I don't know exactly what gracili bacteria are, okay, but in general, there are now sort of a growing list of slight differences in the genetic codes of different organisms. But by and large, what this list is calling the standard code usually works in most circumstances most of the time. So each one of these probably has 1 or 2 differences from the standard code. But it is certainly a larger list than anybody expected when they first wrote this out. They really expected it to be completely universal, no differences ever. But there are a few differences here and there across the phylogenetic spectrum. So if you're interested, you can go to this website and click on these and see, exactly what each difference is. But in general, by and large, as we've been seeing, the differences are usually minor. But there are differences, so that's kind of the important thing. So now we consider it kind of nearly universal, but there are certainly exceptions. Oh, didn't realize I put this in. Okay, so here are some other examples. So we looked at some of these as well. So again, there's some slight differences in yeast and trypanosomes and plants, and nuclear DNA of some of those protozoal type of cells. Okay, but you can get more details at that website. Okay, so far so good? Making sense? We have a triplet code, a redundant code with start and stop, translational start and stop codons that is nearly universal. And another characteristic that they initially thought was always true, but has since shown not to be, is the idea of non-overlapping. And that's this idea of non-overlapping is all about this idea of reading frames. Since we have a triplet code, if I just give you a stretch of letters, a stretch of nitrogenous bases, and just don't tell you exactly where the start is, then you can read it in any of 3 different frames. So if I just gave you that stretch of letters, you could start with the first C, and then you put them in as triplets, and you would get CUC as leucine, AGC as serine, etc., etc. But you could also start at the U. And if you did those triplets, then you get a different set of amino acids, serine, alanine, leucine, proline. Or you could start at the second C, the third letter. And if you do those triplets, you get yet another version. Glutamine, arginine, tyrosine, histidine. So for any given gene, okay, a stretch of letters that code for a product, only one reading frame is correct. So only one of these makes the right, actual protein. So that was sort of the initial idea that people had, was that the code is non-overlapping, meaning that any given nucleotide or any given nitrogenous base is only going to be part of one active reading frame. So only one of these is right. Even though there are three possible, only one of them actually makes the protein. That's the non-overlapping idea. And people started showing that experimentally, that again, only one of the three possible reading frames is correct and makes the right protein. So this seemed to be pretty well established. Until somebody started working with this particular virus called FIX-174. Which is a weird virus for a couple of reasons, but the reason we'll focus on is that it has overlapping genes, which initially weren't thought to be allowed. So we're going to have to add an adverb to this as well. But as you can see in this map, this particular virus has several overlapping genes. So the genes are just given letters. So this E gene overlaps with the D gene, and the K gene overlaps with both the A and the B and the C gene, which is really weird. Right? B gene overlaps with the A and the K gene. So we get lots of overlap within the genes of this virus. So this was, I believe, the first exception to the rule that was discovered. But since then, lots of other viruses, and even most cell types, again, sort of across the phylogenetic spectrum, so plants and animals and fungi and protists, have all been shown to have at least a small number of overlapping genes. So we can add mostly. That might be a good adverb. So it's still mostly non-overlapping, but there's, again, a growing number of exceptions to this rule as well. This is showing this section here where the C and the A and the K gene all overlap. So when people sort of discovered that there were these exceptions, then people thought, well, how come this is the exception? Why isn't this the rule? Because on the one hand, this makes a lot of sense. If I can use each letter 3 times as part of up to 3 different genes like this T is, so that T is part of all 3 genes, as well as that G and that A on either side, you get the idea. That could be really smart. Because I could then theoretically only have to carry around like 1/3 as much DNA. Right? If I use every letter as part of 3 genes, then I can cut the total amount of DNA I have to carry around by 2/3, right? I'd only have to carry around 1/3 as much DNA. So that could be pretty attractive on the one hand, because that's less to copy, it's less to carry around, it's just less to carry around. So why is it still the exception rather than the rule? Since it's not the rule, there must be a downside. So that's kind of the upside. It must be a pretty big downside if this isn't actually the rule, that it's still the exception. So what's a big downside to doing this? In what way? That's exactly what people think. So since that T is part of all 3 genes, if I mutate that, how many genes get affected? All 3. That's probably why this is the exception. So it's bad enough that let's see, this overlaps a lot. It's bad enough that mutating a nucleotide affects one gene, but if they overlap in 2 or 3 locations, then you're messing up 2 or 3 genes at a time, and that's probably really, really bad for, in this case, the virus, but for cells as well. That level of mutation from one, having 3 bad genes now from one mutational event is probably too much of a hindrance to make this the rule. So it doesn't sort of balance out I only need to carry 1/3 of my DNA now. It kind of doesn't balance out as a sort of a cost-benefit analysis. So that's probably the reason. We don't know exactly. We don't really know why, but that's the most reasonable explanation that people come up with, that one change can affect 3 genes, and that's probably really detrimental to the life of that organism. So it does still end up then being the exception rather than the rule, but we are finding more and more exceptions. We are finding more and more examples of overlapping genes. So there is some kind of holdover from kind of this evolutionary experiment. So clearly evolution experimented with it, but decided for the most part not a great idea, but there are still sort of some remnants left over in certain organisms. So we'll call it mostly non-overlapping. So most of the time, each letter, each nucleotide is only part of a single gene in a single reading frame. Does that idea kind of make sense? Okay, so triplet code, redundant code, translational start and stop codons, nearly universal, mostly non-overlapping. And the final major characteristic of the genetic code is, and this one does seem to be always true, is it is collinear, which is shown in this picture. So remember, both DNA and proteins have polarity, right? The 2 ends don't look the same. And there is a collinearity, there's a correlation between the DNA in a 5' to 3' direction and the protein that it encodes in the N-terminus to C-terminus direction. And that's what they kind of show up here. So the information to make the protein is encoded in the DNA in the 5' to 3' direction. And as we'll see, hopefully late next week, when we make proteins, they are made from the N-terminus to the C-terminus, and that's how it's encoded. So the 5' end of the gene encodes the N-terminus part of the protein, and the 3' part of the gene codes for the C-terminal part of the protein. And that always seems to be true. So this collinearity idea is always true. And this then explains essentially why we write the sequences the way we do, right? We always write DNA 5' to 3'. We always write protein sequences N-terminus to C-terminus because of this collinearity aspect of the code. So like I said, as far as we know, there are no exceptions to that particular characteristic. Okay, so that's kind of the basics of the genetic code. Now, before you're all sitting there in fear that I'm going to make you memorize this table, of course I am not. I've gotten through my entire career so far without memorizing the entire genetic code. I know some of it now just because I teach it so often, but I don't know every last 60 of the 64 codons, so I don't expect you to. So if I need you to use it in some way, I will put this picture in the test. Okay? So do not worry about memorizing specific codons with specific amino acids, but do know how to sort of use the table, and I certainly want you to know the 6 major characteristics of the code. That kind of makes sense? Okay. So, like I said, all of this took several decades to work out. So what we've sort of just summarized in about half an hour took several decades to actually figure out. So just kind of realize that as we continue on with this. Okay, so that's how the code sort of works. Now we want to sort of figure out how does this kind of work in the actual life of the cell. So how do we get from this nice genetic code to actually making a protein? Okay, how do we get from the genetic information in DNA to a protein? So that's our next topic, and since I have about 12 minutes, we're going to get started. So the answer to that question is 2 parts. To get from the gene in DNA to a functional protein is a 2-part process. The first part of which is called transcription. So this first slide that is usually referred to as the central dogma of molecular biology, a term which I absolutely hate, but there it is, shows this 2-part process. So we have the information encoded in that double-stranded DNA molecule. We now have a reasonable understanding of sort of how, right? As a triplet code and non-overlapping and universal and collinear and all that stuff. We'll sort of look at it more practically as we go through this. And this part here, this sort of cycle here is DNA replication, so we've already done that. But we're going to focus now on gene expression. Okay, so again, how does this information in DNA eventually end up as a protein? So again, this is going to be a 2-step process, the first of which is transcription, where we're going to take the information for the protein from its DNA form and copy it into RNA. That's what transcription is going to do. DNA to RNA is transcription. Then we'll see that RNA is going to interact with ribosomes and be translated, that's the second part, into a sequence of amino acids that make a protein. Okay, so DNA to RNA is transcription, RNA to protein is translation, and it's, as you can see, obviously a 2-step process. Just like we did for DNA replication, we're going to look at how this works inside bacteria, just because it's ever so slightly simpler than what happens in eukaryotic cells, but the general theme of how it works is the same. And it's just easier to deal with, at this level at least, in bacteria. So we'll do that, just like we did for DNA replication. But realize the broad strokes of how it works in bacteria is the same as how it works in our cells. So this is kind of an overview of the process of transcription. There is going to be, for the most part, only a single enzyme. We're going to need to invoke a couple of other proteins here and there, but the bulk of it is going to be done by a single enzyme called RNA polymerase. So unlike DNA replication, where we had DNA A and DNA B and HU and DNA C and single-stranded binding protein and primates and all those other things, by and large, just really need that enzyme for the most part. So that's going to make this a lot simpler. And we will see lots of similarities between DNA replication and transcription. Except the biggest difference is going to be, of course, DNA replication replicates all of the DNA. Transcription is just about a single gene at a time, basically. But basically what's going to happen is that RNA polymerase is going to locate the beginning of a specific gene and bind to a sequence called the promoter. So that's going to be sort of the signal that here's a gene, here's the start of a gene. And it's going to, instead of copy both strands of the DNA, it's only going to copy one of the strands into a RNA molecule. And we'll have to figure out which strand it's going to do. But this middle part here is really virtually identical to DNA replication, except, of course, we're making RNA instead of DNA. And then there will be some termination signal at the other end of the gene, the 3' end of the gene. And then here's your RNA molecule that you get. So that's, in very general terms, how this process is going to work. And then that RNA, if it's a messenger RNA, will then eventually hop onto a ribosome and be translated into whatever protein that codes for. More on that later. Okay, so this might be a good summary, kind of review slide once we've done the whole process. start with the major enzyme. Again, it is called RNA polymerase. And the bacterial version of the enzyme looks like this. So it has several subunits. So what level of protein structure does this enzyme have? It has multiple pieces. Quaternary structure, right? Quaternary describes multiple pieces having to come together to make a functional protein. So indeed it has quaternary structure. And as you can kind of see from this diagram, it has two alpha pieces, a beta piece, a beta prime piece, an omega piece, and a sigma piece. So the complete enzyme, that's usually referred to as the holo enzyme, is alpha 2, beta, beta prime, omega, sigma. But you can see that sigma is, at least in this picture, is drawn off on the side, because as we'll see, sometimes sigma is attached and sometimes it's not. So when it is attached, we call that the holo enzyme. When it's not attached, so then we just have alpha 2, beta, beta prime, omega, that's referred to as the core enzyme. And what we'll see eventually is that we need the holo enzyme to get the process started. But once it's started, we only need the core enzyme to do the rest. So if we go back to this slide. So to get the process started, we need sigma there, so we need the holo enzyme. Once we get into the later portions, elongation and termination, sigma has left, and what's left over is the core, what's called the core enzyme. And over the years, people have basically worked out what each of these pieces does for the most part. And that's what's summarized here in this table. It just gives you the gene name, the map position you don't have to worry about, the size of the protein if you're interested. What I wanted to focus on in this slide is what each piece does. So the alpha piece seems to be required to put all these other pieces together, and it might interact with some regulatory proteins. The beta piece is the pincer, which kind of lets it stay attached to the DNA, as well as beta prime also forms part of the pincer and has part of the catalytic domain as well. The omega piece seems to help also in assembly of all the pieces, and then sigma, which we'll pay a lot of attention to in a bit, is going to be the major piece that is able to locate, recognize, and bind to this sequence that we're going to call the promoter, which again is going to tell this enzyme where a gene starts, basically. So it's on the front end of a gene. Okay, so like I said, they've kind of worked out what each piece does over the past decade or so. But it did take a while, especially for the alpha and especially omega. We didn't know what they did until relatively recently. So, as you're probably already aware, each chromosome, whether we're in our cells or bacterial cells, whatever, chromosomes have lots of genes on them. So it sort of begs the question, how do we know where a specific gene is and what direction it goes, right? Because we're double-stranded DNA, right? So sometimes the genes are going to be going left to right, sometimes they're going to be going right to left. So sometimes they're going to be encoded in the top strand, sometimes the bottom strand. Some genes are bigger than others. So how do we know where they are? How do we know where they start? How do we know where they end? So there are all these sort of technical, sort of geographic kinds of questions, if we kind of look at this as a map, that need to be answered. So as people start looking at the sequences of genes and what this front end of a gene looked like and the back end of a gene looked like, people started finding sort of similarities no matter what the gene was. So the front end and the back end tend to look really similar no matter what the gene is, so that was obviously pretty important. So what we're going to start looking at next week are these promoter sequences, which define the start of the gene. Where does the gene begin? And will also, as we'll see, be sort of the landing site or the docking site for the enzyme. Okay, so we'll start looking at that on Monday. Okay, so I realize you have a test coming up, so you're probably not going to be dealing with any of this information for a while, and that's fine. But do continue, I hope,